Hello my dear students, hope you all are well. Today I am here to describe the topic transforming principle. Before I am going to start this topic, I welcome you in my channel Eduscope. You know that I have written here Gregor Johann Mendel. His lifespan was 1822 to 1884. So during his lifetime he described that how the characters are moving from one generation to the another generation. That means one generation to another generation to next generation. That is called vertical gene transfer. That is called vertical gene transfer. That means the characters are going down from one generation to another generation. That was described by Gregor Johann Mendel. Next, in the year 1928, Frederick Griffith, he performed one experiment on mice and he told that these genes are moving from the same generation, from one to other, that is called horizontal gene transfer. He stressed that horizontal gene transfer, that means he described the gene of one cell can change the gene of other cell. So that is called transforming principle. Again I am telling you, Frederick Griffith in the year 1928 he performed an experiment on mice and he proved that horizontal gene transfer, so gene of one cell changes the characteristics of gene of other cell in the same generation. That is called transforming principle. So today I am going to show that what experiment was done by Frederick Griffith in the year 1928. Now I am here to describe the experiment which was done by Frederick Griffith. He took one bacteria, that is streptococcus pneumoniae. This bacteria causes disease pneumonia in mice. This streptococcus pneumoniae is having two strains or two types. One is S type, that is virulent, and another is R type, that is avirulent, means non-poisonous. So what is the main difference between S and R? In S, that is having covering, like that. Outside covering is there, that is why it is called smooth. It is polysaccharide. One polysaccharide covers this one, that makes a capsule or covering, that's why it is smooth or called S type. And in case of R type, this type of polysaccharide made capsule is absent. No covering is there, that's why it is called rough, and it is avirulent, means non-poisonous. Now you see, he had taken the two strain of this streptococcus pneumoniae. One is S type strain, another is R type. You see, S type is red in color, I have given for your better understanding, and R type is sky blue color. Now you see, first he has done this experiment. First one, he took S type of streptococcus pneumoniae and injected in the mice. When he took the S type of strain and injected into the mouse, then what is seen? It's seen this mouse is becoming dead. And afterwards, in the blood of this mice, when he collected blood from this one, he got S type of streptococcus pneumoniae in the blood of this dead mouse. Now, it was seen that this mouse was dead and this S type of streptococcus pneumoniae was found in the body of this mouse. In the second case, he took R strain type, that is R type of streptococcus pneumoniae, and injected into the mouse. Then he saw that these remained alive. It was alive. but no R strain is found over here. no R strain was found over here. Is it clear? Next, in the third case, he took some streptococcus bacteria and make them heat kill. By applying heat, this S-type bacteria becomes heat kill. So the polysaccharide what is present on this bacteria is destroyed. So it becomes avirulent. It becomes avirulent, it is injected to the mouse, and see, this remains alive. This remains alive. It is not dead. It remains alive. Okay, in the third case we are seeing it remains alive because it is heat kill. So this S-type of streptococcus pneumoniae becomes avirulent due to heat. Next, in the fourth case, he took heat kill S-type of streptococcus pneumoniae and R-type of streptococcus pneumoniae, and both are injected into the mouse. In the fourth case, he took heat kill S-type of streptococcus pneumoniae and R-type of streptococcus pneumoniae together injected into the mouse. Then it is seen that this mouse, after some times, it is dead. It is dead. Okay. Afterwards, when the blood is taken from the mouse, it has been observed that this S-type of bacteria is found in the blood of this mouse. Now question arises that why in this, when R strain is given separately, it is alive. When heat kill S-type of streptococcus pneumoniae is injected into the mouse, it is alive. But when both are injected, then it is dead and living S-type is found. Frederick Griffith did not explain properly what is the reason of having or what is responsible for changing this R bacteria into S bacteria. Okay, in the year 1944, Avery, MacLeod and McCarthy, these three scientists proved experimentally that DNA of S-type changed the R-type into S-type. Again I am telling you, in the year 1944, Avery, MacLeod and McCarthy explained properly by the experiment that DNA of S-type, heat kill S-type, changed the R-type into S-type. This is virulent. Now I am telling you what is that. This is heat killed S-type. Air covering is not there properly. This polysaccharide covering is lost. That's why it becomes avirulent. 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 Understood? And it is R-type. Now, when both are injected at the same time, this gene remains alive and this gene enters over here, enters into the body of R-type, and this gene of R-type changes into S-type and it regains its polysaccharide covering and becomes virulent and S-type. So that's why this type of S-type streptococcus pneumoniae is found in the dead body of this mouse in the fourth case. I think it is clear to you. Thank you very much for being with us for a long time.